So we're going to continue with our anaerobic lecture, and we're going to start out by talking about some of the anaerobic systems. Now, we already talked about isolating our anaerobes and using special primary and selective anaerobic media. Now, how are you going to incubate these cultures after you inoculate them? So you need to generate an anaerobic environment for these organisms to grow. And generation of an anaerobic environment can be done in one of numerous ways. One way is through an anaerobic chamber, or what's called the glove box. Another is using a jar technique, such as these special gas pack jars that I'll show you pictures of, as well as bio bags. So the anaerobic jars or pouches, these are what are going to commonly be used in a clinical microbiology laboratory. So you have a gas pack system that usually is a cold reduction system that contains a palladium catalyst in it. And that palladium catalyst is going to, usually you're going to add some sort of water to it, and it's going to generate a gaseous mixture of 10% hydrogen, 5% carbon dioxide, and 85% nitrogen. And in these systems, there's always an oxygen reduction indicator, and that we already talked about is your risazorin or methylene blue, which is going to turn pink or blue if there's been oxygen exposure. So here on the top left you see an anaerobic jar. So what you do is you inoculate your plates. You stick your plates, stack them up in this jar in the right direction. So usually your micro plates are incubated upside down. These jars come with an anaerobic environment envelope that you usually have to add water to, and you stick that inside the jar. And then there's the indicator strip, the risazorin and methylene blue, and there's a little area that you slide that into the jar as well. You then tightly put the lid on and screw it down. There's usually a gasket on the lid so that no oxygen can get in. Now the minute you open up that indicator strip, it is going to be pink or blue, because the second you open it up, it's going to be exposed to oxygen. However, once you let it sit in the enclosed jar, that pink or blue color will go away as all the oxygen is removed in the jar. Now, there's also pouches, and these anaerobic pouches, you can see it on the right hand side, right bottom, are very similar to the microaerophilic Campylobacter pouch. So if you were working in a lab and you rarely did an anaerobic workup, or you only had a couple of plates, you might use the pouch system. So the pouch system is just like the Campy microaerophilic system, where you put your plates in the bag. It comes with a little liquid plastic little container. You pour all of the liquid, squeeze all of the liquid into this little slot into the plastic area that goes all the way down into this sachet in the pouch, and when the liquid goes in, it generates the gas and removes all the oxygen, and you have to close that bag very nicely. There's a special system to close the bag, like a clip. Now, whenever you're using these types of systems, you put them in the incubator right away and you let the gas do its thing and remove all of the oxygen. Now, when you go to take these jars or pouches out of the incubator, the first thing you want to do is look at the indicator strip. If the indicator strip is pink or blue, depending on what indicator you're using, you don't even bother looking at those plates. those plates have been exposed to oxygen throughout their incubation, and even if there was an anaerobe present in the specimen, you've killed it. So you would have to request another specimen. So you always want to look at the indicator strip first before you open that jar, because the second you take the clip off the pouch or undo the top of your anaerobic jar and oxygen starts to get inside, the indicator strip will turn color. So you always want to look at the indicator strip before you open them. So here is an anaerobic jar with the little pouch that contains the chemicals in it to generate your gas, and you see the little blue indicator strip that's in the clip right on the side of the jar that you could see it. You want to face that indicator strip out in the incubator so that it's facing you. So when you open that incubator the next day, the first thing you see is that indicator, and you want to see that it is a white. It shouldn't be blue or pink. Again, your pouch or bag systems, and this shows you the little plastic liquid with the clip. Now it looks like a campy pouch to me, but the anaerobic system is exactly the same as the campy. Usually the anaerobic bags have blue writing on them, and the campy bags have red writing. Now, if you were doing anaerobic research or working specifically in an anaerobe laboratory, you might have a glove box. And this is actually an entire chamber, a gas-tight chamber or hood that has glove portals. So you don't actually handle the specimens like you would a typical specimen. You're handling them through gloves in this gas-tight chamber. There's a special entry area where you put your materials inside the chamber. There's a hydrogen-containing atmosphere circulating throughout a palladium catalyst inside the chamber to constantly remove oxygen. So here is a glove box type of anaerobic chamber, and you could see this is more of a scale. It looks very plasticky to me. I'll show you some others that are more commonly used. But your specimen and your plates are introduced into the environment through an airlock, and then you would lock the airlock back up, and then you would let the chamber remove any oxygen that came in, and then you would start working with your specimen. This is a gloveless anaerobic chamber. So you would actually stick your hands through, and you're actually touching the specimens just like you would normally. And there is the catalyst that's constantly removing oxygen from the atmosphere. Here's another glove box where you would stand up and put your hands through the gloves and look into it, and everything would go into the chamber from that airlock on the right hand side. And again, here's someone actually working in a glove box. Now we're going to talk about the anaerobic culture criteria. So all specimens that are submitted for anaerobic culture must also be aerobically cultured. I've already said that a couple of times. Now the minimum conditions that you could use are inoculating your specimen onto 5% sheep blood agar and incubating it in the CO2 incubator. You can also inoculate blood and McConkey agars and incubate those in an ambient air incubator, in addition to inoculating your anaerobic media and putting your media in anaerobic conditions. Now, the observation time for anaerobes is 48 hours for primary culture. So you're usually not going to open up your anaerobic jar for 48 hours. You're going to look at it, make sure the indicator is not blue or pink after 24 hours, but you're not actually going to open it and look at your plates for 48 hours. If you have growth on your anaerobic media at 48 hours, you should gram stain that organism growing on the plate to see what you might be looking at. Also, you're going to do your typical biochemical and antimicrobial susceptibilities on any isolated anaerobe, just like you would for any other organism that you isolate in the clinical microlaboratory. Now, there are criteria that you can follow to prove that you actually have an anaerobic organism. So if you take each colony and subculture, so 2 of the same colonies, and subculture them to 2 plates of the same media, you can put your differential discs on your media. We'll talk about some of these differential discs. And you stick one of the plates in a CO2 or carbon dioxide incubator and the other in the anaerobe jar. If your subcultured organism grows better anaerobically than in CO2, it should be worked up. Because keep in mind, there are some anaerobes that will grow very lightly in CO2. An obligate anaerobe won't, but just a general aerobe can grow a little bit in a CO2 incubator. So if you get more growth of the same organism when it's incubated anaerobically than when it's incubated in a CO2, that would be an indication that it is truly an anaerobe. So if you inoculated an organism onto a blood agar plate, the top row, BAP, in an ambient air incubator, if you had growth there, it would be indicative of an aerobic organism, or possibly a facultative organism, because facultative organisms can grow anywhere. You shouldn't get a capnophilic or an obligate anaerobe growing in an ambient air incubator. If you have chocolate agar, an organism inoculated on chocolate agar and incubated in a CO2 incubator, you would get your anaerobe your facultative organisms growing, you'd get your aerobe growing, and a capnophilic organism could grow, but not an obligate anaerobe. Now, if you take your media and your organism and incubate it anaerobically, your aerobes and most of your capnophilic organisms will not be able to grow anaerobically, but your facultative organisms and your obligate anaerobes and your just regular anaerobes are going to be able to grow anaerobically. So there's a little asterisk that Haemophilus influenza will grow anaerobically on Brucella blood agar, but you're going to have better growth of that organism when it's grown on chocolate agar and in a CO2 atmosphere. So remember, Haemophilus influenza requires factor X and factor V, and it usually is going to be inoculated onto chocolate agar and incubated in a CO2 incubator. But the Brucella blood agar does have the right factors in it, and Haemophilus will grow lightly. So for identifying your anaerobes, it's important to gram stain them. It's important to look at the morphology of the organism growing on a plate. It's important to look at how it grows on the selective media that you've inoculated it onto. It's also important to do your biochemical analyses of your organism so that you can identify it. So like the other organisms we've talked about, some of the other organisms where you might drop a disc on your plate and then incubate it, and you're looking for those zones of inhibition, you are going to do that with your anaerobes. So let's say you have growth on your primary anaerobic media. So since it's not selective, you don't really know what the organism is. You're going to do a gram stain on that organism. If it's a gram-positive cocci, you will streak out your organism onto another plate, and you're going to drop an SPS and a nitrate disc on it. If it's a gram-positive bacillus that is either branching or dipteroid looking, remember the Clostridium, I keep saying Clostridium, Corynebacterium. Your Corynebacterium organisms, those dipteroid organisms, if it looks like that, you're going to drop a nitrate disc on it. If it looks Clostridium-like, and we'll talk about Clostridium, these are usually pretty large gram-positive bacilli. Those don't need any disc. If it's a gram-negative cocci, you will drop a nitrate disc. If it's a gram-negative bacillus, you can drop a VKCl nitrate and bile disc onto that, and that's going to help you identify what organism you might be dealing with. Now, not all clinical labs drop discs onto plates because we have our instruments that we can put our growth into, and the instruments will do a lot of these tests for you and tell you what you might have. So for example, you have a wound specimen, some sort of aspirate from a tissue wound. Now, in this wound specimen, there are multiple organisms, because remember, usually anaerobic infections are polymicrobic. So you have Escherichia coli, you have Bacteroides fragilis, which is an anaerobe, you have a pigmented Prevotella, which is another anaerobe, you have Staphylococcus aureus, which we've talked about in the past, and you have another anaerobe, the Peptostreptococcus anaerobius. So you have 3 anaerobes, and you have 2 facultative organisms. So you are going to inoculate 2 different sets of plates and incubate one set in the CO2 incubator and the other set anaerobically. What are you going to see when you take these plates out of the incubator? Well, on a blood agar plate that you incubated in a CO2 incubator, of course you're going to see the E. coli and the Staph aureus. On, MacConkey agar, you're only going to see the E. coli because MacConkey agar is selective for gram-negative organisms. The PEA, you're only going to see the Staph aureus because PEA selects for is more specific for gram-positive organisms. Columbia CA's Columbia agar, you can get your E. coli or your Staph aureus growing on that. For your anaerobic plates, for your pre-reduced Brucella blood plates, all 5 of these organisms are going to be able to grow on your Brucella blood plate in an anaerobic environment. Your pre-reduced PEA plate, you're going to get your Bacteroides fragilis and your pigmented species of Prevotella. You could also get your Staph aureus and your Peptostreptococcus, because the PEA will allow your gram-negative, Bacteroides is gram-negative rod, your Peptostreptococcus is a gram-positive organism. So PEA will allow gram-negative and gram-positive anaerobes to grow. But for facultative organisms, the gram-positive organisms grow a little better on the non-reduced PEA. Your pre-reduced BBE agar, which is a selective media for Bacteroides, of course only the Bacteroides is going to grow. And then your pre-reduced KVLB anaerobic media, your Bacteroides fragilis as well as your pigmented Prevotella will grow. So if you have a specimen and you grow them both in an aerobic and anaerobic environment, you may see multiple organisms growing, and you have to think about which media you inoculated, what atmosphere, what organism grew better on which plates. So your E. coli and your staph will likely grow better in the CO2 than anaerobically, but they will be able to grow on some of those plates anaerobically. Now your discs, we already talked about dropping some of the discs. So there's the V or the vancomycin disc, there's the K or the kanamycin disc, there's the C or the colistin disc, and these are antimicrobial agents. So if you drop these discs on an anaerobic organism and it's susceptible to vancomycin, so in other words there's this zone of inhibition around a vancomycin disc, it's variable for kanamycin, and it's resistant to colistin, meaning it grows right up to the colistin disc. It might be a Clostridium species. If it's resistant to kanamycin, it might be Porphyromonas. If it's resistant to vancomycin, kanamycin, and colistin, grows right up to all of them, it might likely be a Bacteroides fragilis organism or a Prevotella. So Bacteroides fragilis is known for being highly resistant to antimicrobial agents. So these discs will give you an indication of what you might be looking at. You could do gas liquid chromatography also on some of these organisms if you were in a very specified anaerobic laboratory. You may not do this type of test in a typical clinical microbiological laboratory. So here's a schematic diagram for presumptive identification of anaerobes. So you have organisms on your anaerobe plate. If you gram stain that anaerobe and it's a bacillus that has a spore, you're likely looking at a Clostridium species. If it's a gram-positive bacillus with no spore, it might be Actinomyces, Bifidobacterium, Bacterium Lactobacillus. Remember, we talked about Lactobacillus previously. Lactobacillus is a facultative organism. It can grow in CO2, it can grow in anaerobic atmosphere, or Propionibacterium. And there are lactobacilli species that are considered anaerobes. Now, if it's a gram-positive coccus, it is likely Peptococcus or Peptostreptococcus. If it's a gram-negative coccus, it is likely Veillonella. If it's a gram-negative bacillus, it could be Bacteroides, Fusobacterium, Porphyromonas, or Prevotella. So this is a slide that you should memorize because it is telling you which genera are what type of morphology. So that's important, because if you're looking at an exam question and it's a deep wound aspirate from someone that had a trauma, you should automatically have your light bulbs going on and off saying, "Ooh, this might be an anaerobe." And if it then says the organism was isolated from a Brucella blood agar grown in an anaerobic atmosphere and it was a gram-negative bacillus, you should automatically be thinking of those 4 organisms. If the answer choices for that question were Bacteroides, Peptococcus, Lactobacillus, and Clostridium, you don't even have to read the rest of the question. The only answer was the gram-negative bacillus, the Bacteroides. All the others are not gram-negative bacilli. So that's how you have to think when you're reading some of these exam questions. Now, your frequently encountered anaerobes are Clostridium, Bacteroides, Fusobacterium, Prevotella and Porphyromonas, Veillonella, and Peptococcaceae organisms. And here is just a pie chart showing the clinically significant anaerobes. So in a clinical microbiology laboratory, when you do get an anaerobic infection, most commonly, the one you are going to see the most is going to be a Bacteroides species. The next would be your gram-positive cocci. The next would be your Propionibacterium, your Clostridium and some other organisms, and then Fusobacterium. So without a doubt, Bacteroides causes the most anaerobic infections. Now, we already mentioned some virulence factors. Some organisms can produce toxins. For example, Clostridium botulinum and Clostridium tetani produce neurotoxins, very potent neurotoxins. Clostridium perfringens produces an alpha toxin or lecithinase, and this is considered a lethal toxin. And then Bacteroides fragilis produces capsules. We're going to move on to the third part of the lecture where we're going to discuss specific anaerobic organisms.